we are going to restart. Are you all there? Are you all there?
Yes.
I'm talking to myself. Okay. So now we are going to start looking at morphological changes that involve the early body pattern formation. As we learned in the last lecture, during development of a living organism, after cleavage division, you have to come across the pattern formation, and pattern formation involves two things, which is basically the body axis formation, and the second important part in the body pattern formation is the germ layers formation. Both of these things are very deeply rooted in early embryo, or rather I would say the oocyte. When mothers in Drosophila, they lay down the egg is already defined. The body axes are partly set up in the oocyte, which is followed once fertilization takes place. Then these body axes, which are partly defined in embryo, in early egg, they are then established in pattern in the early embryo. Here you can see along the anterior-posterior axis, already the egg, which is laid by the mothers, it comes already in defined anterior and posterior axis and dorsal and ventral axis. On the anterior side, it is defined by, or morphologically, you can see the micropyle end, and posterior end is more round. And this then through pattern formation, it undergoes, at cellular blastoderm changes where we see the ventral endoderm. So if we have a cross section of the fruit fly embryo, if this is the cross section, if I draw it like this, so this will be the mesoderm, and then this is going to be the ventral ectoderm, which is here in the yellow, dorsal ectoderm, next one, and then amnioserosa. This is basically extra embryonic tissue. We'll talk about this later on as well. And already by this stage, you have very clear parasegment or sign of segmentation in fruit fly embryo. Now, as you already have seen, there are in fruit fly, you will have AP axis, anterior posterior axis, and you will have DV, which is dorsal ventral axis formation. This is anterior posterior body axis formation. In case of dorsal ventral axis, this is divided into 4 regions, which we just saw. So this is the dorsal, this is the ventral, and this is divided into mesodermal tissue, which is going to make muscles, internal connective tissues, ventral ectoderm, which is going to be largely nervous system, which I just explained you as a formation of neuroblast. So neuroblast cells emerge from this region, and the dorsal ectoderm, which contains extra embryonic tissue, which is the amnioserosa membrane. Now let's look at the anterior-posterior body axis formation in fruit flies. So anterior-posterior body axis formation, as I told you, they are the oocyte or egg cell comes in partially defined body axis formation, or its oocytes axes are already defined, which lay down the foundation for embryonic anterior posterior axis formation. If you disturb this loose definition, loosely defined anterior and posterior oocyte axis, you will destroy the whole developmental plan. It is that important. So how we know this? In early part of 1970s, Kristin Nuslin-Follard and Eric Weishaus, they both were postdocs in Walter Gehring's lab, and they were interested in understanding how segmental body plan is established, how segmentation takes place. Okay, so this is the embryo. We just saw these large-scale movements, the morphological movements. Okay, now we are moving into molecular part. They decided to set up a genetic screen. They said, okay, let's do a genetic screen and see the defects in body segmentation. And if we see defects in body segmentation, we take these phenotypes as our readouts. If we are able to see disturbances in anterior posterior body axis formation or dorsal ventral, etc., it means we are disturbing the genes which are responsible for developmental plan or body axis formation or pattern formation, etc. It was one of the most successful screen in Drosophila, and I think after Morgan's discoveries of chromosomal theory of inheritance, if I think fly has contributed something enormous in the field of biology, is this screen. What they came up with when they observed after genetic screen, and it was simple. So you feed the males, fruit fly males, with EMS. EMS, as you know, is a mutagen. The flies which will hatch, so these males will be then crossed with normal females, and then the progeny which will hatch from this cross, in the progeny you will look for phenotypes which highlights defects in body plan. Okay, as I told you, it was a Herculean task. It was a huge genetic screen. What they did, they classified the genes, the phenotypes they discovered, into 5 different categories. The first category they named as maternal genes. And why they named maternal genes? Because they found that, you know, if mothers, they are carrying mutation for a gene, and we cross this with a wild type male, we will see a specific phenotype. But if we have normal wild type mother and cross it with a mutant male, we don't see phenotype. So it means Mutation, when it comes from mother, we'll see effect, and they call them maternal effect genes. Then they've found another class of mutants in which phenotypes was like, you have normally we see these embryos, their size, they are of specific defined size, then the segmentation coming up. What they saw that these embryos were smaller than the normal, and there were big gaps in the segmentation. Segments were not at all there. Many gaps in the form of absence of many segments or deformities. Then another class of genes they named later on when they discovered those genes that they are pair rule genes. And finally, there was a class of genes discovered from this successful screen, which were named segmentation genes. And eventually, the last class was selector genes. We'll talk about all of them one by one. They are listed here in an order, and they were not discovered in such an order because the molecular one, they started understanding their molecular function in next 20 years. But what was clear that there is a cascade of gene expression. Maternal genes are the genes which in the oocyte. So when mother lays the egg, mother deposits messenger RNA for these genes like this. And these messenger RNAs, they act as, for us, as definition of anterior axis and posterior axis. And one of the very famous gene is bicoid. And later on, we'll talk about nanos, caudal, etc. Bicoid messenger RNA is already deposited by mother on the anterior side. And if you lack bicoid in mothers, you know what happens? You are headless. Head is not developed at all. So, as I said, the anterior-posterior body axis is already defined in the oocyte, which is then later on undergoes changes and establishment of clearly defined anterior-posterior axis of the embryo. Now, here you saw the consequence that if you don't have messenger RNA deposited by mother, your head is not developed, you're dead, okay? So we'll see how these changes undergo. Now, these, just like bicoid is deposited here, they discovered that on the posterior pole, also there are specific messenger RNAs like nanos and caudal, and and these messenger RNAs are anchored. They are specifically present with cytoskeletal structures there, and these messenger RNAs, they never get translated unless fertilization takes place. Now, I said it's a cascade of gene expression. So when these messenger RNAs, the anterior and posterior maternal genes, get translated, they lead to activation of a class of genes which we know today as gap genes. And this is the first zygotic transcription. Immediately after fertilization, up until certain hours, and I can't recall the specific minutes, or maybe 1 hour or so, we don't have zygotic transcription. We don't have zygotic genes being transcribed. These messenger RNAs which are deposited by mother, their proteins, they act on the nuclear genes and then activate. Hunchback is one of the zygotic gene, not only zygotic, but also it's a maternal effect gene as well. Gap genes are transient as well. So maternal genes, bicoid, nanos, caudal, they are transcription factor. They are transient, they activate gap genes. Gap genes are also transient. Before they disappear, they activate another class of genes which are called pair rule genes. And as a result of pair rule genes, you see very clearly defined 14 parasegments established. Even skipped and Fushithrasu are two genes which are which represent pair rule genes. Now, these by this stage, when you have uh, protein products of bicoid, gap genes, and pair rule genes, you know, the idea or the beauty in development is that you are one single cytoplasm with many nuclei, and all these nuclei are sharing the protein products of maternal genes, of gap genes, and early stage pair rule genes. When you have pair rule genes, you are already in segmentation, and parasegments are defined, but these just before the onset of segmentation, when all the nuclei are sharing the same cytoplasm, they all are exposed to, they all are sharing these transcription factors. Pair rule genes, when they finish establishing these parasegmented body plan and they disappear, then they activate segmentation genes. Segmentation genes are mostly cell signaling genes. Here, like wingless, hedgehog, engrailed, DPP, and so on. Now, this is very important developmental stage. Nuclei are no more sharing transcription factors. Each nucleus is now basically independent cell through the process of cellularization, and only communication between two adjacent cells or two adjacent layers of cells in different compartments is through these signaling molecules. And eventually we see selector genes activated, and these selector genes are like Hox genes, abdominal A, abdominal B, or etc. Selector genes are basically master regulators which give specific identity. They specialize. They are involved in specialization. Here you have cell fate determination has taken place, and here these genes eventually are the last step in the specialization of different cell types. As I already told you, in the oocyte, you have maternal genes, and maternal genes are already expressed in the ovaries. So when we look at the within the ovaries, so let's say I just quickly explain you. When one of this stem cell is going to be, selected as so these are the primordial germ cells. They are not yet undergone. They are not yet in meiosis. They are still diploids. One of them moves into meiotic phase. It divides. This is, let's say, the stem cell hub. And then out of four product of meiosis, one gets selected as oocyte. The others are, the others develop as nurse cells, polyploids. They express messenger RNAs. These are polyploids. Their job is to nurse, to feed messenger RNAs into oocyte. By the end, and this is through different developmental stages in the germarium, so you have one nucleus or one cell, which is this one, which is the future oocyte. Now, at this stage, within the ovary, at the time of egg formation, you have messenger RNAs which are deposited at different axes, and they define the axis of the embryo. Bicoid and nanos caudal are deposited in specific position, and then eventually this egg is laid. It comes out of the females, They lay them on the food or in fruits, and this egg has these bicoid and nanos caudal, the maternal genes, which are shown here, which I already explained you. As I told you, these are just messenger RNAs, and there are nearly 50 maternal genes which define the anterior and posterior body axis formation. 50. I just named few, I think, nanos, caudal, bicoid, Oscar, etc. Now, as soon as the fertilization takes place, the translation of these messenger RNAs take place. Okay. And then they contribute to clearly defined anterior and posterior body axis formation. However, in further investigation of the maternal genes led to three different classes of genes within the maternal genes. They found through the mutant phenotypes that there are maternal genes which only affect the anterior region. And there are maternal genes which only affect the posterior regions. And then there are maternal genes which affect both terminal anterior and posterior regions. Here are the cartoons which shows you representation of genes which affect only the anterior region. Here, this is the wild type embryo. This is how the anterior body axis look like. This is the posterior in wild type. So on the anterior, you have acron, head, thorax, abdomen, and telson in the embryo. This class of mutant which shows defects only in the anterior region, you see this was the phenotype they saw. The posterior was fine. The middle part of embryo was fine. The anterior was missing. The acron was missing. Then they found that the posterior was missing in another class, which is the second one. And the example was the nanos. They found the nanos mutant defects are only in the posterior side, but not on the anterior. And bicoid is the example for anterior class of mutants in maternal genes. And then they found torso mutant. It showed mutant phenotype in both anterior and posterior. Both acron, telson were missing in torso mutant. Now, what are the molecular functions or features? Why bicoid affects only the anterior side and nanos only the posterior side? Yeah, each class, the anterior mutants or the posterior mutants, or they act independent of each other. Nanos caudal do not affect this phenotype. Bicoid mutant does not affect this phenotype, and torso acts independent of both bicoid and nanos. As I said, after fertilization, the translation of these messenger RNAs, which are deposited by mothers, it only takes place after fertilization. If you take unfertilized egg, you will find these messenger RNAs localized precisely at the anterior side, at the posterior side. And they will remain untranslated in the absence of fertilization. Only after fertilization, you will see this. Now, how do we know this? Last time we talked about few tools, and if I say differentiate between unfertilized egg and fertilized egg, or show how that what I'm saying is correct, that bicoid or nanos, they only get translated after fertilization. You can design a simple experiment. You will take fruit fly embryo and you will take antibody against bicoid, as well as you will do in situ hybridization. In situ hybridization will tell you, okay, messenger RNA signal is lying there. If you do immunostaining using anti-bicoid antibody, you won't see any protein in unfertilized egg. And immunostaining will only show you, because immunostaining means you're using antibody against bicoid protein. Antibody is against protein, not against messenger RNA. So in situ will be showing you the presence of messenger RNA, but immunostaining will show you, okay, there is messenger RNA, but it is not being translated in unfertilized fruit fly embryos, which is shown here. I'll go back to previous slide in a moment. So this is your in situ hybridization, which shows presence of bicoid deposited by mothers. And this is the immunostaining, which shows you protein, very high concentration of protein, and then you see a gradient. So this is, red one is a signal of bicoid messenger RNA, right just in the anterior side. And if you look at the protein, very high concentration, and then the concentrations goes down because there is a concentration gradient. It's a one big cytoplasm. Protein is freely diffusing. It's free to diffuse in the one big cytoplasm. Now, they did some very interesting experiments. And once they knew that bicoid mutant is the interior, it affects only anterior region, they came up with the idea that it means there is something special in the anterior region. And experiments were, I would say, very intelligent, but very simple. And I would say an intelligent experiment is always simple. What they did, they pricked the embryo, and because they knew already the wild type phenotype, they knew the bicoid mutant phenotype, okay? And what they did, they said, okay, let's take just the cytoplasmic fluid and inject it into the bicoid mutant. What they are doing, they are not doing any genetic manipulation. Just with a fine needle, take fluid, inject it into bicoid mutant. And what they saw, some anterior structures develop. It means there's something special in this cytoplasm. And they had no idea by that time. Okay? This messenger RNA, all these things came later on. Now, the next experiment was even more interesting. I said, okay, let's take this wild type fluid, inject it in the middle of the embryo. And what they found, that this transfer of fluid from wild type into the middle of embryo resulted in head-like structure in the middle of embryo. It means the cytoplasm of anterior region contains something. And with the discovery of messenger RNA, which I just told you, they could then conclude what is that special. And once they cloned bicoid gene and all antibodies against bicoid, they could show that, yeah, already in the unfertilized egg, we have bicoid messenger RNA, and we can clearly differentiate between unfertilized and fertilized eggs. In unfertilized, we will only see the messenger RNA, and in fertilized, because this bicoid messenger RNA only gets translated after fertilization by the sperm. And then later on, it turned out that bicoid is a transcription factor. It's protein. It has binding sites in DNA of critical genes, which later on we came to know as gap genes. And translation of this messenger RNA of bicoid is crucial for activation of zygotic genes. Otherwise, zygotic genes never get translated. Okay? And we see big defects. Now, after bicoid, they turn towards the posterior region genes. What is special in posterior region genes? And similar to bicoid, they discovered, oh, there's very special class of genes, and they discovered 9 different factors, at least 9 different factors. Some of them are, so for example, nanos, caudal, Oscar, etc., which are deposited on the posterior side, in the oocyte. And after fertilization, their messenger RNAs get translated. And they somehow counteract the effect of bicoid from anterior side. So on the protein from nanos caudal, after fertilization, they also diffuse like it is shown here. So this is the immunostaining, you see. If you will do in situ hybridization, you just like bicoid, you will see a localized signal. But after fertilization, you see, you can lay down a concentration. If this is the y-axis, this is the x-axis. On the x-axis, you have anterior, posterior. Now, what do you have on the early stage embryo? Let's say this is the embryo. This is anterior side. This is posterior side. And these are the concentrations of, on this y-axis, we have bicoid. On this y-axis, we have nanos. So what we have is a concentration gradient visible by this, and a concentration gradient of bicoid. And there's another maternal effect gene, which is called hunchback. Hunchback protein also gets translated, but there's no gradient. It's uniformly distributed like this, which is shown here. Now, the point is, what is the role of nanos? They discovered that this uniform concentration of hunchback is counteracted by nanos. The nanos protein, it results in a gradient of hunchback. Initially, hunchback is uniformly distributed, but after fertilization, the hunchback protein is restricted to initial half of the embryo. Okay, and if you have deletion, if you don't have hunchback in mother, so if mothers are mutant for hunchback, nanos become dispensable, because nanos function was to restrict hunchback in a gradient like fashion. Once hunchback is absent, nanos remain dispensable. Now, how, you must be wondering how these messenger RNAs, they remain localized. How? So there are other factors. For example, one is mentioned here. Oscar is a factor which localizes nanos messenger RNA just here, okay, in the unfertilized egg. Similarly, there are other factors which are responsible for maintaining bicoid here in the messenger RNA here in the anterior side. In addition to nanos, caudal was another posterior effect. Similar to nanos, it is present on the posterior, but caudal messenger RNA, it's just like hunchback, it's uniformly distributed throughout the egg. However, just like hunchback is restricted by nanos, if I draw caudal here, caudal is also messenger RNAs like this. Now, when fertilization takes place, what we see high concentration of bicoid, and this bicoid actually results in a gradient of nanos, caudal. Caudal, which was messenger RNA, was uniformly distributed like this. The protein is restricted by action of bicoid. And if you have mutation in caudal, we see abnormal abdomen, so a posterior phenotype. So now, what is happening if we try to draw the embryonic molecular picture? Molecular picture is immediately after fertilization, the hierarchy of gene activation takes place as a result of uh, translation of these maternal genes. Bicoid, it gets translated, it restricts spread of caudal protein into the anterior side. Nanos blocks spread of bicoid towards posterior. So at the end, what we have a gradient of nanos and caudal, and we have a gradient of bicoid like this. And then we have hunchback. Now, all of them are transcription factors. They are freely diffusing in the cytoplasm, and the nuclei which are sharing this syncytial blastoderm, they are being exposed to these transcription factors, and they activate the zygotic genes in those nuclei. Now, as a result of these gradients, now we have uh, not just bicoid and nanos gradients, we have very clear bicoid gradient, nanos gradient, hunchback gradient, which is due to the nanos, and then caudal gradient. Caudal gradient is due to action of bicoid. Now, here you see the genetic locus of hunchback gene, and hunchback is not only maternal effect gene, but also zygotic gene is activated by, so these are the bicoid binding sites in the DNA in the promoter region of hunchback. This is the transcription starts at this arrow. At -100, -200, -300, you have many bicoid binding sites. They activate hunchback gene expression. Now, as I said earlier, these are transient. They are not there forever. They set up the stage for the zygotic genes and the activation of hunchback, Krupel, NEPs, etc., which later on they discovered that, oh, these genes belong to the class of mutants we defined as gap genes. And you remember I told you, if let's say Krupel is missing, a big gap in the embryo will be out. So Krupel is normally expressed here. Now, if Krupel will be mutant, embryo will be much more smaller than the normal. And these transcription factors, again, their concentrations are in very defined discrete regions, and nuclei in those, so all the nuclei in this region, for example, they will be exposed to Krupel, and these ones will not be expressed to Krupel. Now, you have to think this embryo, early embryo, forget about gap genes at the moment. This, if I lay down embryo here like this, high concentration of bicoid, hunchback, nanos, caudal, etc. Now, think of having nuclei here. A nucleus which is exposed to very high concentration of nanos, caudal, etc., and least concentration of bicoid, what is happening to it? There is a kind of gene expression pattern at very, very early embryonic stage. Foundations of a gene expression, cell type specific gene expression pattern is being laid down here. High concentration of bicoid will bind to not only hunchback, but to hundreds of other genes. And then a low concentration of bicoid, which is, let's say, here or here, as compared to this region, these nuclei will be committing to different fates in the long run. So we have very early on different kinds of gene expression patterns being laid down, which set the stage for cell fate determinations later on in development. So after gap genes, you have these, they activate the pair rule genes. Pair rule genes then activate the segment polarity genes, and segment polarity genes, before they disappear, engrailed is, for example, one of the segmentation genes. Fushi tarazu, even skipped, etc. These are the pair rule genes. Even skipped, Fushi tarazu, hairy, runt, odd, etc., which contribute to very, very early signs of segmental body plan. And remember, what was the question? Question was how body X's are formed, how pattern formation takes place, how segmental body pattern is established in flies. And we are getting answer to those establishment of segmental body plans in the form of genes which play a role in that segmental body plan. Now, what we already learned is that, structures on the anterior achron, the telson, we already saw that the bicoid mutant loses anterior side, the nanos mutant, the posterior side, and torso on the both side. Now, what is the torso doing? We have already covered two classes, which were anterior effect gene, posterior effect genes, and then the final one is torso. Now, torso is uniformly present throughout the membrane of the fertilized egg. The molecular function we learned much later when they start characterizing the torso. Now, torso messenger RNA is present in the form, this blue one is uniformly distributed all over the cytoplasm in the embryo. However, the phenotype is only on the two extremities, the anterior and posterior, the terminal mutants. Why? What is special there? What they discovered that after fertilization, when the torso, maternal torso, is translated and torso protein is distributed throughout the fertilized egg plasma membrane, the ligand for torso is present only on the two extremes. The torso receptor, it undergoes cleavage, although torso protein is uniformly distributed, but torso protein undergoes cleavage in a very localized manner. So not torso receptor, the ligand. I apologize for confusion. So the ligand for torso, which is a fragment of protein called trunk. So this trunk is processed only in the anterior pole or the posterior pole. And this ligand, after cleavage of trunk, is available for the torso receptor only in the, and torso receptor is actually, which is product of maternal torso gene, is a receptor, a cell membrane receptor, which receives this ligand, torso ligand, and then leads to activation of downstream cascade of gene expression pattern. That's why when you have torso mutant, you will find phenotype, the consequences only on the two anterior or posterior axis. Is it clear so far? Any questions? So we stop here. Next time when we meet, we'll talk about, instead of anterior posterior axis formation, we are going to talk about dorsal ventral axis formation. Today is Thursday, and we are going to have a recitation. Do you remember all? So we'll talk about some of the tools, 6 to 7, and at 7 I have the advisory board meeting. Yeah, Fariha.
Okay, sir, I have a question. First, you mentioned that we have a factor that's called Oscar, and it holds or it restricts the nanos to the posterior end. So my question is that Oscar a protein, like it's holding it in the form of the protein, or it's itself a messenger RNA that's present in the posterior part, and that's holding, or
Yeah,
it's
very
somehow
good
holding
question. So
the
there are
nanos.
three things there. There is Stauffen, there is Oscar, and Stauffen and Oscar messenger RNAs are there as well. And one of them, I forgot, is a non-coding RNAs. And they are linked with, I think that's the microtubules, if I, or yeah, microtubules. Stauffen is the protein, and I can't recall if Oscar is a messenger RNA. I remember Oscar messenger RNA. Whether it's a non-coding or no, I have to look back.
Okay. I had another question
Yeah.
that I forgot.